It's 2 o'clock, and we're supposed to start now. So thank you all for coming. I see Cody's family, Cody's grandma and cousin, all the way from Virginia Beach. Thank you. I have to say today is a very special, Dr. Doge, special day for me, definitely for Cody. and I remember when I first came here, I came here early 2012 for an interview, and Cody was one of the students set up to have lunch with me. Adriana, I remember you too. You came to have lunch with me. And at that time, I was fascinated by Cody's Southern accent. And I realized if I were to come to EVMS, I must have him in my lab. And sure enough, that happened. And I have to tell you that why is Cody so special, actually for a lot of people in the audience too, is talking about research. I feel like Cody's passion for research is stronger than my passion, compared with him. That he would subscribe, using his own student stipend to subscribe to journals. When I was a student, I wouldn't do that. I would just steal somebody's journal to look at. And also, honestly, his seminar today is not going to cover every single thing that's done in the lab. I'll give you one example. The stuff he's not gonna talk about today is how he used CRISPR to knock out a gene of interest in our lab. And to me, that was a big deal. That work actually led to publication of our paper earlier this year, primarily driven by a postdoc. But that piece of study wouldn't be complete without Cody's contribution to use CRISPR. Many of you have heard of that technique to modulate gene expression. And also, Cody is one of those guys, you know how some people like me, I just do what I need to do on campus and I leave? That's not Cody. He does what he needs to do, and he does more. For example, all the fundraising event. And that's why he was honored by the school a year or two ago for his philanthropy work. And anytime you want anything, people know. Say you have a fridge broken, or you have something heavy you have to lift, just get Cody. He'll be there. So he's like a firefighter. And with a heavy heart, let me conclude by showing you this. So this is Cody evolution. Okay. From 1986 to 2018, I have only witnessed this little part here. Transition from here to here. Okay? But I was told by his family that Cody's transition from here to here was even more amazing. The only thing I can say is I look forward to Cody's future. I know Cody's future is bright, like sunshine. And let me remind you, today we have a reception at 4 p.m. in Lester Hall, room 210, upstairs. Thank you.
All right, thank you, David. Can you guys hear me? Yes, okay. All right, so thank you all for coming. The title of my dissertation is TTF1, Thyroid Transcription Factor 1, Modulation of Intercellular Communication and Intracellular Signaling. So just to give you an outline of my presentation today, I'm going to give you some background on lung cancer and thyroid transcription factor 1. Then I'm going to go into the data. So the hypothesis for my dissertation is that TTF1 can alter intercellular communication and intracellular signaling in lung cancer, and this can alter lung cancer biology. And we use 3 different aims to test this hypothesis. Aim 1 was to elucidate the mechanisms of how TTF1 can sensitize cancer cells to chemotherapy drugs. Shown there. No, I don't like that one as much. There we go. Aim 2 is to investigate the impact that exosomes have on angiogenesis and if TTF1 can modulate this effect. That's right up there. And aim 3 is to determine if TTF1 can alter gap-junctional intercellular communication between cells. And then I'll summarize the findings and then go into future directions on where our lab is headed now. So a little bit of background on lung cancer. Lung cancer is a leading cause of cancer-related deaths both in the US and the world. It has a very low 5-year survival rate of only 15%. And just like they say, cancer is not one disease, but a cluster of 200 separate diseases, lung cancer can be further divided into different subtypes. Roughly 15% of lung cancer is referred to as small cell lung cancer, and 5% are referred to as other. These are the rarer types of lung cancer, like lung carcinoid tumors, which are of neuroendocrine origin. The vast bulk of lung cancers are non-small cell lung cancer, shaded in blue, which can be further subdivided into adenocarcinomas, 40% of lung cancers, squamous cell carcinomas, which are 30%, and large cell carcinoma, which is 10%. So my talk today won't really talk about smoking, but I get asked this question a lot. How much does smoking contribute to lung cancer? Roughly 10% of lung cancer patients are never smokers, so 90% have a history of smoking. The reason smoking is so bad is because it causes a lot of mutations in the genome. And that's what really cancer is. It's a disease associated with mutations in the genome. And there are many genes that are mutated in cancer. The most common mutation in lung cancer and all cancers is P53. P53 is the proverbial brake that is applied whenever there's aberrant cell growth. And this makes it a tumor suppressor. When it's mutated, it gets turned off. But tumor suppressors are just one type of gene mutated in cancer. Other genes are called oncogenes, and when they're mutated, they become active. Roughly 15% of lung cancers have an activating mutation in EGFR. That's epidermal growth factor receptor. And what that is, it's a receptor tyrosine kinase that activates several downstream pathways which result in aberrant cell growth. And these pathways include AKT or KRAS. KRAS itself is mutated in about 30% of lung cancers. It's the most common oncogene mutated in lung cancer. What's interesting is even though EGFR and KRAS share some pathways, they're mutually exclusive in cancer. You don't get lung tumors with mutations in both. The original thinking behind this was, well, if there's shared pathways, you don't need to mutate the others. There's no selective pressure. But work done by the Harold Varmus lab at the Memorial Sloan Kettering has shown that when you insert mutated active KRAS and EGFR into the same lung cancer cell, it overwhelms the cell and it dies. So these are two big players in lung cancer. But the most important one is TTF1, and that's what we'll be talking about today. So TTF1 stands for thyroid transcription factor 1. It's also called NKX 2.1, designating it as a homeodomain containing transcription factor. It's a transcription factor, which means it turns on and off other genes, and it's a homeodomain containing protein. That's its DNA binding domain. Homeodomain proteins are known for their role in morphogenesis, and TTF1 is no exception. TTF1 is essential for lung development. Mice homozygous null for TTF1 die at birth due to poor lung development. And like any good developmental gene, it's also involved in cancer. And in fact, for years, pathologists have been using it as a marker for specific types of lung cancer. TTF1 is expressed in about 70% of lung adenocarcinomas and about 90% of small cell lung cancer. So just by doing the math, over 40% of lung cancers are TTF1 positive. So interest in TTF1 being associated and a factor driving cancer really began about 11 years ago when our lab and others were investigating an event referred to as the 14q13.3 amplification. So what that is 14 is chromosome 14. Q is the big arm of the chromosome. P is the small arm. Q is the big arm. And in this big arm, there's a region called 13.3, which becomes focally amplified in about 15% of lung adenocarcinomas, driving tumorigenicity. When our lab was studying the genes that could be involved in this, they found that TTF1, in concert with other genes, were able to enhance cell proliferation, and siRNAs targeting TTF1 attenuated this effect. So this set TTF1 up as being a gain-of-function oncogene, and that got people excited into studying it. Since that finding, however, they found new roles for TTF1, including many tumor-suppressive features that it has. So this kind of gives TTF1 the interesting moniker of being a conditional oncogene, meaning that there are different contexts where TTF1 can be oncogenic or tumor suppressive, and that really depends on the genetic background. Case in point, in EGFR mutated cancers, TTF1 is oncogenic, while in KRAS mutated cancers, TTF1 is tumor suppressive. However, TTF1 overall expression does correlate with a favorable patient prognosis in lung cancer. Patients with TTF1 positive lung tumors have a better outcome than those that are TTF1 negative. So with that in mind, we were kind of curious how TTF1 can alter intercellular communication and intracellular signaling in lung cancer, and how this could affect the different hallmarks of lung cancer, including angiogenesis, which means blood vessel formation, and chemotherapy drug sensitivity, which now leads us to AIM-1. So a few years ago, a paper by the group of Jeff Whitsett looking at TTF1 biology showed that transfecting A549 lung cancer cells with TTF1 sensitized them to cisplatin, a very common chemotherapy drug. But this was never followed up on, and we were kind of curious. We saw this effect, what could be mediating it? So for our AIM-1, we're going to look at how TTF1 can sensitize these cells to chemotherapy drugs, and AIM-1A is to look into that effect. What is happening inside the cell that's causing the sensitivity by TTF1? AIM-1B is to see, well, how common is this? The generality of TTF1 sensitivity to chemotherapy. Will this happen in other lung cancer cells? And AIM-1C is looking to see if there's an extracellular component that could be mediating this. We're very interested in intercellular communication. Is there a secreted factor that could then be coming back to the cell, sensitizing it to cisplatin? So our work for AIM-1 has been put together in a manuscript that we submitted to Scientific Reports. We're finishing up the reviewers' comments, and we should be resubmitting it pretty soon. So look for it. So the first step, of course, is to transfect our cells with TTF1. We chose A549 cells. It's one of the most common lung cancer cell lines used for drug sensitivity. It was also the cell line used by Jeff Whitsett's article. So we want to see if we can recreate that finding. We also used H460 cells, another lung cancer cell. So both these are KRAS mutated lung cancer cells, but we wanted to see about EGFR mutated lung cancer cells. So we used H1975 cells and transfected them with TTF1. As an additional control, we also transfected these cells with a mutated form of TTF1 called TTF1 HDD. HDD stands for homeodomain deleted. That's its DNA binding domain, and that's how it acts as a transcription factor. Once this is removed, it can no longer bind to DNA, and it should be functionally silent. So we transfected these cells with these constructs, and we also did immunofluorescence to make sure that TTF1 is being expressed and is localized in the nucleus here in these A549 TTF1 cells, as they are. So we did these drug sensitivity assays. So this is just a dose response curve. There we go. So the y-axis is cell viability. The x-axis is increasing concentrations of cisplatin. What we found when we take our three cell lines and we introduce increasing concentrations of cisplatin, A549 cells expressing TTF1, this red line, has an increased drop in cell viability compared to the controls. So using our software to calculate IC50, that's the point where 50% of the cells have lost their viability. We again see that TTF1 expressing A549 cells are more sensitive to cisplatin. So we repeated that with our H460 cells and saw a similar effect. But like I said before, TTF1 has been shown to be tumor suppressive in KRAS mutated cancers, and these are both KRAS mutated cancer cell lines. What about EGFR mutated cancers? So we repeated this experiment with our H1975 cells. We saw the exact opposite effect. TTF1 makes these KRAS mutated cancer cell lines sensitive to cisplatin. It makes this cell line resistant to cisplatin. So what could be mediating this? This is kind of what our whole point of AIM-1A is, to figure out what's going on inside the cell. So we went through the literature, did a lot of PubMed searches, and we decided to focus on three different genes. AKT, glycogen synthase kinase 3, both alpha and beta, they're two homologous proteins with 97% sequence homology, and beta-catenin. All major proteins involved in cancer, and also been shown to be involved in chemotherapy sensitivity. Importantly, a lot of research has looked at these three genes and how they modulate cisplatin sensitivity in A549 cells. So we're going to go through it. I'm going to go ahead and show you the mechanism first because there's a lot going on. There's a lot of negative regulation. I also have it drawn up here too, so you can refer back to it. But in resting cells, quiescent cells, there's a lot of active GSK3, glycogen synthase kinase 3. And beyond just the basal level of activity, there's also a phosphorylation that can occur on a tyrosine, which makes it more active. In these cells with active GSK3, GSK3 can go to beta-catenin and phosphorylate it, marking it for destruction. Through this phosphorylation, TTF1 or beta-catenin can be degraded by the proteasome via the axon beta-catenin destruction complex. However, in proliferating cells like cancer cells, there's a lot more active AKT, which can be seen with a phosphorylation. Phosphorylated AKT can go to this GSK3 and inhibit it with a phosphorylation on a serine residue. Once that happens, beta-catenin can then be activated and go to the nucleus, where it can transcribe its target genes, which result in drug resistance. This includes genes like MRD1, multi-drug resistant protein 1, or survivin, very conveniently named. Also, a couple years ago, it was even shown that active phosphorylated AKT can go directly to beta-catenin and activate that, which also results in it going to the nucleus and transcribing its target genes. So we began our study looking at beta-catenin. Beta-catenin has many different functions on the cell, depending on where it's located. So we used different compartments to figure out which beta-catenin could be modulated by TTF1. And we found that on NA, total beta-catenin was reduced. However, nuclear beta-catenin, which is where it has its drug resistance effects, was statistically significant downregulated by TTF1. Cytoplasmic beta-catenin didn't have much of an effect. We were curious, too, to follow up on this, and we used confocal microscopy with the help of Dr. Latanzio, and we detected beta-catenin with IF. What we found was that in our control A549 cells, that we see a lot of nuclear beta-catenin localization. However, in our cells expressing TTF1, we see mostly membrane-bound localized beta-catenin. This makes sense. Beta-catenin is known to be also a focal adhesion protein, which complexes with E-cadherin, a known target of TTF1, and I'll talk more about E-cadherin later. So we moved up the pathway and started looking at GSK3. So these are just the representative Western blots detecting the GSK3 protein and its modifications. And what we found was that in GSK3 alpha, there is a reduction in that inhibitory serine modification phosphorylation. While for GSK3 beta, we saw not only an increase in total GSK3 beta, we saw an increase in the tyrosine phosphorylated GSK3 beta. That's the excitatory modification. So this shows to us that TTF1 expressing cells have more active GSK3. So we wanted to, of course, modulate GSK3 to see its effect, and we used a drug called lithium chloride. Lithium chloride is a known inducer of beta-catenin, but its actual mechanism of action is to increase those inhibitory serine phosphorylations on GSK3. And when we treated our A549 cells, empty vector and TTF1 with them, we saw that in our A549 TTF1 cells, lithium chloride treatment was able to increase the resistance to cisplatin. In our empty vector cells, we saw a slight increase, although not significant. This is probably because in our TTF1 cells, we have a lot more active GSK3, not so much in our empty vector control cells. So therefore, there's less active GSK3 to inhibit. We also saw this effect with similar GSK3 inhibitors as well. So moving up the pathway, we're now looking at AKT. What we found was that while total AKT expression did not change, the active phosphorylated AKT was greatly reduced by TTF1. We also looked at PRAS40, which is a known target of AKT, and that was also downregulated. And that also occurs in the H460 cells as well. Now, the H1975 cells, which if you remember became resistant to cisplatin after TTF1 treatment, showed the opposite effect. Phosphorylated AKT went up. So we were curious as to how AKT could be playing a role in this sensitivity. So we treated our A549 cells with MK2206. It's an AKT inhibitor. And you can see that it was able to wipe out active AKT, as well as reduce the inhibitory GSK3. And what we found was that in our empty vector cells, we're able to sensitize them to cisplatin after MK2206 treatment. For A549 TTF1 cells, we didn't see an effect. Again, similar to that with lithium chloride, A549 TTF1 cells have very little active AKT, so an inhibitor is not going to have much of an effect on these cells. So, as I said before, we're very interested in intracellular communication, so we're kind of curious if there could be an extracellular factor that could be playing a role in this. So we set up an experiment called the conditioned media experiment. So we took our cells, A549 empty vector or A549 TTF1, and we plated them out, added fresh media, and then we waited 48 hours. During this time, the cells will grow, but in the process, they'll be secreting all sorts of factors into the media: proteins, extracellular vesicles, cytokines. And then, after that 48 hours, we'll collect that media and then set up for a drug sensitivity assay. So we'll drop in our cells into this assay and then add our conditioned media. And with this, we can play around. It can either be empty vector cells getting the empty vector conditioned media or getting the TTF1 cell conditioned media, or the opposite. So we want to see how this conditioned media affects this response. Then add cisplatin, and then after 48 hours, we'll see cell death. And what we found was that for A549 TTF1 cells in orange and red, the conditioned media source had no effect. They killed the cell at the same rate. However, in our empty vector cells, we find that blue is the TTF1 conditioned media, and it can actually make these cells more sensitive to cisplatin than the conditioned media that's from the empty vector cells. So there's a lot in the conditioned media, whether it's proteins, whether it's extracellular vesicles, and we kind of want to whittle down a bit. What could this be? So we chose to look at exosomes. So what are exosomes? I'm sure you guys have heard of them by now. I know I've talked enough about them. These are small vesicles that are released from cells. They then go to the recipient cell and change how that cell behaves. And it does this through the delivery of bioactive molecular content. And almost everything in terms of category of biomolecules has been present in there. RNA, proteins, all sorts of transmembrane proteins, even DNA. And they can alter the recipient cell behavior by altering gene expression through the delivery of RNA, delivering the protein, or engaging receptors on that recipient cell. And through this, they've been shown to play key roles in cancer. This includes the induction of the epithelial to mesenchymal transition, which is linked to metastasis, enhancing proliferation, or also angiogenesis and drug resistance, which we'll be looking at today. So we chose to do this cell viability assay, and this time added in our exosomes, but first we characterized them. So we took our exosomes, we harvested them through ultracentrifugation, and then we probed them for CD9 and extracellular vesicle marker. And this is our soluble fraction, so we were able to pull these extracellular vesicles out. And then we used the nanocyte to characterize them. The nanocyte is an instrument that uses a laser and Brownian motion that's predictable for the vesicles of this size to kind of determine their size, to make sure that these are in the range that we're looking for, around 100 nanometers. And also gives us our concentration so we can use a dose response for it. So we added these exosomes to our cisplatin sensitivity studies, and we didn't see much of an effect. And when we did a dose response, we also didn't see an effect. So this tells us that cisplatin sensitivity is not really conferred by anything in the exosome fraction. So we then used that soluble fraction, which I showed you before, and repeated the experiment, and we were able to see the same effect. In our TTF1 expressing cells, there wasn't much of a difference between the source of the conditioned media. But for our empty vector cells, receiving the TTF1 cell conditioned media made them more sensitive to cisplatin. So this is kind of what we're interested in. We have some candidates that could be mediating this effect. But to summarize, AIM-1, TTF1 is able to sensitize KRAS-mutated cancer cells to cisplatin, and this is found to also be with a decrease in the phosphorylation of AKT. However, in our EGFR-mutated cancer cells, we see the opposite. TTF1 makes them resistant to cisplatin, and we see an increase in the phosphorylation of AKT. When we took our cells and modulated AKT, GSK3, and beta-catenin, we're able to alter that sensitivity to cisplatin. And we're also able to see sensitivity to cisplatin in the conditioned media. And I didn't have time to show this today. When we did these experiments with other chemotherapy drugs, we didn't see this effect by TTF1. So with this and our findings with the different conditions with the cells, makes us believe that these findings can be used for further research to guide treatment of lung cancer patients. So now that leads us to AIM-2. So we're going to actually have some positive effects with our exosomes. So a little bit of background. An article we published a few years ago showed that the exosome-depleted conditioned media from our A549 TTF1 cells was less angiogenic than that from our control A549 cells. So kind of just show you the mechanism of what happened. A549 cells are capable of secreting VEGF, which will engage the VEGF receptor on the endothelial cells. These are HUVECs, human umbilical vein endothelial cells. And what this occurs is this activates that cell and stimulates angiogenesis. However, A549 TTF1 expressing cells also secrete another cytokine called GM-CSF. GM-CSF will stimulate that endothelial cell to secrete a soluble VEGF receptor. And what this does is it sops up all the VEGF, making it unable to engage the VEGF receptor on the endothelial cell. So that's what we published in this article here. However, the role of exosomes is being investigated later, and that's when I took over. So AIM-2, we want to see the effect that these exosomes have on angiogenesis. And that's AIM-2A. AIM-2B was to determine if TTF1 can modulate this effect. And AIM-2C was to characterize the protein cargo with mass spec and see if there's any changes in the constituent proteins. So in order to test for the angiogenic potential of these exosomes, we did an in vitro tube formation assay. So this is just how it's set up. You have a well and a layer of matrigel. It's a 3D matrix meant to mimic the extracellular matrix. And you add our endothelial cells to them. Now, if the environment that they're in is angiogenic, you'll see the creation of these networking patterns. It's a honeycomb pattern. These branches come out, and it's very angiogenic. In contrast, if the environment is not angiogenic, these cells won't create these networks. So when we did this experiment with these endothelial cells treated with different exosome preparations, we found that in our A549 control cells that they were able to have an increased angiogenic potential to the endothelial cells. However, when these endothelial cells were incubated with exosomes from A549 TTF1 cells, there was a reduction in the angiogenic potential of these exosomes. So in order for blood vessel formation to occur, these endothelial cells have to migrate through the extracellular matrix, and they have to be motile. So we used a Boyden chamber assay to test this. So how this is set up is you have a well that's split in two by a filter. This filter has very tiny holes, smaller than the cell. And what this does is you drop on your endothelial cells in the top chamber. In the bottom chamber, you have a chemoattractive environment. This is an area where the cells want to migrate to, if they can. So we add our cells, we added our exosomes to the top, and then we let it go overnight. If these endothelial cells are motile, what you'll see is they're able to push through these little holes at the bottom and adhere to the bottom of that filter. And this is what it looks like here. And if you want to test for motility, you count the number of cells at the bottom of that filter. When we did that, we saw that there's a reduction in the migrated cells when they were incubated with exosomes from A549 TTF1 cells. So we showed that these A549 TTF1 exosomes confer less of an angiogenic potential and less of a motile phenotype to our endothelial cells. This was not what we were expecting. So we previously published that our exosomes from A549 TTF1 cells have increased VEGF cargo inside them, VEGF being very angiogenic. But we found kind of the opposite phenotype with them. So we decided to investigate their protein cargo with mass spec. And with the invaluable help of Dr. Dutta and Dr. Niall Whitty, we performed our analysis on the cargo proteins. And TTF1 changes a lot of the proteins in these exosomes, but what really stood out is MUC5AC. MUC5AC is a mucin protein, that's involved with many aspects of tumorigenesis, as all mucin proteins are. And that was the most differentially regulated protein in our exosome fraction by TTF1, so that's where he chose to focus on. We then followed up on that study with an ELISA looking at MUC5AC to confirm that, yes, indeed, it is downregulated in these cells. And we went back to the cell. So A549 TTF1 expressing cells have almost absent MUC5AC expression. When we did an ELISA looking at our A549 cells, we see that for every 20 micrograms of cell lysate, there's about 1.5 nanograms of MUC5AC protein. And at the qPCR level, we also see a reduction in MUC5AC as well. So MUC5AC, again, is really interesting because it's also been shown to be involved in angiogenesis. Beyond that, other aspects of tumorigenicity, including migration. And by using other cells, we saw that TTF1 is able to downregulate MUC5AC in many different cell lines. We were curious as to what could be happening in the patient. So this is data from the Director's Challenge Lung Study. It's a patient database. What we found was that TTF1 and MUC5AC expression are negatively correlated. We then looked at survival estimates. How could this be used to predict the survival outcome of a patient? And by ranking these with TTF1 high, low, MUC5AC high, low, we're able to see that patients that are TTF1 high, MUC5AC low, which is this top red line, have a better survival estimate than those in the green line, which are TTF1 low, MUC5AC high. So this can be used to predict patient outcome. So to summarize the AIM-2, the exosomes from our A549 TTF1 cells are less angiogenic than those from our control cells, and TTF1 alters the exosome cargo. MUC5AC was very downregulated in our exosome fraction, as well as the cellular level, and lung cancer patients show a negative correlation between TTF1 and MUC5AC. Furthermore, TTF1 positive MUC5AC lung cancer patients have a higher survival probability than the opposite, those that are TTF1 negative, MUC5AC positive. So this is just one type of regulation. We're also interested in gap junctional intercellular communication. And the reason for that is our lab and others have shown that TTF1 directly regulates the proteins E-cadherin, which makes up the adherens junctions. These are the ones that hold the cells together, as well as occludin and claudin. Occludin and claudin, which make up the tight junctions. So these proteins involving in junctions are also very tumor suppressive. You see lots of these junctions with increased tumorigenicity. But these are just two types of cell junctions. We were kind of curious if TTF1 could be a master regulator of cell junctions. And we're curious as to what effect that TTF1 could have on gap junctions. So gap junctions consist of a ring of 6 connexin proteins that are embedded in the plasma membrane. And one ring from one plasma membrane will connect with one of another membrane and couple together to create a continuous channel between them. And there are about 21 different types of connexin proteins in the body. An example is connexin 43, which is the most ubiquitously expressed and the most well-studied. And what these gap junctions do is that it allows the passage of small molecules between cells. This can include glucose, different ions, cyclic nucleotides, microRNAs. It's even been shown that peptides can be threaded through these gap junctions. As opposed to adherens junctions and tight junctions, gap junctions are kind of a mixed bag in terms of their role in cancer. They've been shown to have tumor suppressive and oncogenic functions. It really depends very specifically on which type of connexin is being involved. It also depends on the cell type or the stage of the cancer. For the most part, connexin proteins and gap junctions have been shown to be tumor suppressive. They can ratchet down cell proliferation, but they also tend to be very metastatic. Overall, however, in cancer, you do see a reduction in gap junctions and gap junctional intercellular communication. So we were curious, okay, TTF1 alters these junctions. What about gap junctions? So AIM-3A was determined if TTF1 can alter gap junctional intercellular communication between cells. AIM-3B determine if TTF1 can modulate the expression of the connexin proteins that make up those gap junctions. So in order to test for gap junctional intercellular communication, we did what's called the parachute assay. So we take two populations of cells. Our recipient cells, we add DID. It's a lipophilic dye that stains the membranes. Right. These are our recipient cells. Then we have another population called donor cells. What we do is we treat them with calcium AM. Calcium AM is a small molecule that can passively enter the cell through the cell membrane. However, once it's inside the cell, it can no longer exit the cell the way it came in because it becomes metabolized by intracellular esterases. This process also makes it fluorescent. It'll fluoresce, it's very green. We then will take our donor cells and drop them onto our recipient cells. So just think of this as like a 50x magnification of this. And then we let them incubate. During this time, they'll set up these gap junctions. And if cells are in close contact with each other and gap junctional communication is in place, you'll see these neighboring cells start to turn green because they're receiving this calcium AM through these gap junctions. That's what it looks like there. So what we're looking for then are these double positive cells, recipient cells that are also positive for calcium AM. And this is how we measure to see the amount of gap junctional intercellular communication. And when we did this and measured through flow cytometry, we found that, well, there was very little gap junctional intercellular communication to begin with. This isn't a surprise. Tumor cells already have low gap junctional intercellular communication. However, what we did find is that although it is low, we do see reduced GJIC in our A549 TTF1 cells. So which genes could be having this effect? So this is work that was done by Caitlin Helmsley, who is a research tech in our lab, and she did a PCR array looking at the expression of different cell junction proteins. And this is set up to where she used an immortalized lung cell line called BASTB cells. And with treatment with doxycycline as the TET-on system, she can turn TTF1 on. Without this doxycycline, TTF1 is not turned on. So she used the TET-on system, added doxycycline or not, and then harvested the RNA from them and ran this PCR array. And what we found was that proteins like E-cadherin, CDH1, occludin, and many of the claudins were upregulated, which is exactly what we already knew. However, GJA1, gap junction associated 1, also known as connexin 43, was downregulated. So connexin 43 stands out because it's the most ubiquitously expressed protein in cancer, most ubiquitously expressed connexin protein. It's also very associated with cancer, so that's where we chose to focus our study. So I followed up on this, and I took our BASTB cells and our A549 cells, and I tested connexin 43 expression. And what we found was that TTF1 expression reduces that of connexin 43. When we took our H441 cells, which are TTF1 positive, and knocked TTF1 down with shRNAs, we see a de-repression in the expression of connexin 43. Furthermore, we also saw this at the protein level as well. A549 and BASTB cells expressing TTF1 have a loss of connexin 43 protein. So to summarize this part of AIM-3, while our A549 cells have low intercellular communication between them, it still does occur through these gap junctions. However, when A549 cells are expressing TTF1, there's a reduction in the connexin 43 protein and a reduction in the gap junctional intercellular communication. Furthermore, our data from the Cancer Genome Atlas and Director's Challenge Lung Study show that TTF1 and connexin 43 are negatively regulated in lung patient tumors. So this kind of led us to another investigation in how intercellular regulation can alter each other. So we're kind of curious if gap junctions can alter exosome secretion and vice versa. So these are two major forms of intercellular communication, but right now, if they regulate each other, hasn't been investigated until we showed up. So this is a very zoomed-in version of the gap junction. So gap junctions work by being in a gap junction plaque. That's where you find these connexin proteins. That's where you find this intercellular communication. However, outside of the plaque is a region that has been termed the perinexus. This is where you get those hemichannels in that one plasma membrane. They're uncoupled, so they're continuous with the extracellular environment. In this perinexus, this hemichannel is tethered to the cytoskeleton through a protein called ZO1, which binds to the C-terminal tail of connexins. Since connexins and gap junctions are involved in many different disease processes, drugs are actually being made to modulate this effect. A peptide called alpha-CT1 has been developed, which mimics the C-terminal tail of connexin 43. And what this does is it then displaces connexin 43 off of ZO1, allowing it to transfer to the gap junction plaque. Once there, it can couple with a hemichannel from the adjacent cell and can enhance gap junctional intercellular communication. So we use this alpha-CT1 to modulate our gap junctional communication and see what effect this had on exosome secretion. So after treating our cells with these drugs, we found that alpha-CT1 treatment resulted in less harvested exosomes after ultracentrifugation compared to the PBS control or the reverse peptide, which is the same alpha-CT1 but flipped. So this is kind of a thing that we're really interested in, beyond just TTF1, how this intercellular communication regulates each other. But to summarize, this is the 1,000-foot view of today. What we found is that in EGFR-mutated cancer, TTF1 increases the phosphorylation of AKT and results in cisplatin resistance. However, in our KRAS-mutated cancers, TTF1 reduces the phosphorylation of AKT and also results in cisplatin sensitivity. So these lung tumors are very high in MUC5AC and mucin expression. What we found in our control cells is that it can release exosomes that are positive for MUC5AC, and these exosomes are also pro-angiogenic. However, when TTF1 is involved, this expression of MUC5AC is suppressed, and it releases exosomes that are MUC5AC negative, and they're also anti-angiogenic. In our cells, we see gap junctional intercellular communication. However, when TTF1 is expressed, we see a reduction in the expression of connexin 43 protein and a reduction in the gap junctional intercellular communication between these cells. So where things are going to go now, future directions, we want to look for a combinatorial effect between cisplatin and statin in our TTF1 lung cancer cells. I didn't show this, but Michael Lai, a postdoc in our lab, has found that TTF1 actually increases the efflux of cholesterol out of these cells. That will then make them very sensitive to statins. Statin treatment kills the A549 TTF1 cells. So we're curious if there's be a synergistic effect between statins and cisplatin in these tumors. We also want to look into the extracellular factors that could be mediating the cisplatin sensitivity by TTF1. And we also want to look at exosomal MUC5AC and angiogenesis. MUC5AC is involved in angiogenesis, but we want to be able to link these two proteins, or MUC5AC, with our angiogenic potential of these vesicles. And we're also looking in the gap junctions and exosome regulation of each other. So there's a lot of people I like to acknowledge, of course, the Moo Lab, Michael and Lauren, the two postdocs, Caitlin Helsley, our tech, Nicole Cox, our master student, as well as our med students, Chris, Kevin, John, Blair, and Kay, as well as Shelby and Megan, our biomedical sciences students. I'd like to thank the members of the committee. They've been very helpful. Of course, David Moo, my advisor, Dr. Kirscher, Krishna, and Sims. I'd also like to thank Dr. Niall Whitty and Dr. Detta for their work with the mass spec. I'd like to thank Dr. Kim and the flow cytometry facility, Dr. Latanzio for his help with the confocal microscopy, Jess and Eric, my unrelated family members, thank you. My family, thank you. Adriana, Aziz Sinkar, who was one of our collaborators for AIM-1. And last but not least, I'd like to thank this group of people here. I don't know exactly what we were trying to convey, if we were trying to look tough or something in this picture. Here's one of us being slightly more normal. But with that, I'll take any questions. Thank you. Oh, Dr. Godfrey. Mm-hmm. Mm-hmm. Yes.
My question is, in a patient, would you have both cells that do express TTF and those that don't? In other words, would there be a source of inflammation from tumor cells like
Mm-hmm. Are you asking about intratumoral heterogeneity to where you have mixed TTF1 expression in this tumor? You do, but this would be more of a kind of a gain of function sensitization. So whether all the cells would have to express TTF1 or just a subset of them, we wouldn't know until we look into the patient.
I guess what I'm getting at is that the potential therapy
Mm-hmm.
whatever those factors are.
Mm-hmm. Mm-hmm. Yeah, yes, yeah. Brooke? Thank you, that's a good question. So our work was on KRAS or EGFR mutated lung cancer cells. Those are the most common oncogenes that are mutated in cancer. Not all of the lung cancers have these. So we did work with a lung cancer cell line at H1437 cells, which are mutated in neither. What we found was that TTF1 did not change the sensitivity to cisplatin in these cells. Furthermore, we didn't see a change in the phosphorylation of AKT either. So, yes. Are you asking for like animal models with TTF1 expression? Well, one common animal model is the KP model. That should be the Tyler Jacks mouse model. And they have done work with TTF1, but they don't look for drug sensitivity. They look for metastasis. But that would be a good model to use in terms of drug sensitivity because they've shown mixed TTF1 expression in these cells. Interestingly, this is kind of how the anti-metastatic role of TTF1 was originally somewhat elucidated, was that they found in this KP model, it's a very famous model, that the metastatic lesions were all almost universally TTF1 negative, while the parent tumors did have a little bit higher a lot higher TTF1 expression. So, yeah, Dr. Morse. Which slide? Mm-hmm. Oh. We can just call it the 6 because there were 6 of us. Yeah. Yeah. There we go. Right. Any other questions? Oh, Kevin. Oh, thank you. Mm-hmm. In their A549 TTF1 positives, I mean, beyond AKT, TTF1 has been shown to also positively regulate P53 expression. We have not looked at that, but that is something that has been shown to be in play. Manon? Don't say congratulations yet, but hmm Okay, actually, thank you. We did use gemcitabine and carboplatin, and they had no effect. And this kind of goes to our finding that we believe that this data can be used to drive patient research. TTF1 sensitized in cisplatin for KRAS mutated cancers, but it didn't do it for like gemcitabine. So therefore, with patients with TTF1 positive lung cancers, cisplatin might be a better course of treatment than gemcitabine or carboplatin. Yeah. Oh, well, of course. Yeah, I mean, chemotherapy drugs have a lot of side effects. Hair loss, cachexia, nausea. Yeah, so cisplatin and gemcitabine, cisplatin and carboplatin, carboplatin and gemcitabine are very common combinations used to treat non-small cell lung cancers. Yeah. So, yep.
Okay, so
Oh, yeah.
the next stage is, well, thank you for all. And we have to go into closed door session so we can grill Cody, right?
Yeah.
And make him suffer.
Right.
But before we go into that, I'm sure folks won't mind that Cody used selfies to take a photo.
So we like to take a lot of photos in our lab because we have a lab folder for it. Let me see. All right, so here we go.
Maybe we should steal the people in the audience.
All right. Everybody ready? I don't think it's working. No. How many PhDs does it take to use a selfie stick? All right, ready now? Okay, thank you. Yeah. Thank you, David.
Anyway, thank you.
And there's a reception at 4 o'clock as well. So, and there's cake with my face on it. I saw that before coming here. So.
Yeah, reception 4 p.m. Lasper Hall 210. And I think as a committee member, maybe we sit here. We have to. Yeah, thank you. Good job.